Hello, everyone, and welcome to Nucleai Academy. We're so glad you could join us for today's talk titled AI Spatial Biomarkers and Diagnostics for Antibody-Drug Conjugates, or ADCs. My name is Nilima Kumar. I'm the head of marketing at Nucleai, and I'll be your moderator for today's session. So before we dive in, I'd like to go over a few quick housekeeping items to make sure we have a smooth experience. The session today will run for approximately 60 minutes. So it'll be a 45-minute presentation followed by a 15-minute live Q&A. The entire session will be recorded, and we'll be sharing the recording with all the registrants after the webinar, so you can also watch it on demand at your leisure. All attendees are going to be muted, and this is just to avoid background noise. So if you do have any questions for our speaker during the live Q&A, you can use the Q&A box at the bottom of your screen, and we'll do our best to answer as many questions as possible during the Q&A section. Now, to keep the session interactive, we'll also be launching some poll questions throughout the talk. We want to hear from you, so please keep an eye out for those. They will pop up on the screen, and you can take just a few seconds to respond. And we'll also share the results of the poll. And finally, if you're experiencing any technical issues, you can just first try refreshing or rejoining the session. But you can also message us in the chat again at the bottom of the screen, and our production team will do our best to help. Now, without further ado, I'm excited to introduce my colleague and our expert speaker for today, Dr. Jason Reeves. Jason is the Director of Application Science at Nucleai, and he works very closely with our biopharma customers, as well as our in-house pathologists, data scientists, and engineers, where we apply a suite of AI models to imaging and pathology data and extract biomarker and MOA-related insights for accelerating drug development. Jason is not new to the field of spatial biology. Most recently, he's been here from the early days. He was the head of computational biology at NanoString, now Bruker Spatial Biology. And he was also a translational research scientist at Jounce Therapeutics and Sanofi earlier in his career. So he's got the great mix of the technology side as well as the pharma side. With that, I will pass the ball over to you, Jason.
Thank you so much, Nalima, for that kind introduction. And thank you all who are joining us today. Really glad to see such a great crowd come out and join us for this webinar. And we're really excited to talk to you about some of the technology that is really shaping the next steps that some of the therapeutics in oncology, and now potentially even moving outside of oncology, are taking. So as mentioned, we're going to be focusing on spatial biomarkers that are AI-powered and AI-driven. And throughout this webinar, we're going to be talking about the evolution of how we can further create and model key components of the mechanism of action of ADCs. Now, as the therapeutic market has evolved, we've gone from drugs that have been effectively untargeted chemotherapies through continual evolution to more and more targeted therapies. That included therapies that were driven towards genomic biomarkers and specific mutations that were present within patients, to immunotherapies, and ultimately now to multispecifics and ADCs as the next generation of therapeutics that have direct targeted mechanisms of action that allow drug companies to design therapies that are really specific to an individual patient tumor. And this evolution has been one that should be coupled not just with evolving drug landscape, but it also demands that we think about how these drugs work, the context in which they work, and how we can better capture that type of information for patient selection and understanding their overall response patterns in the clinical trial setting. So we're going to talk specifically about ADCs. I do want to point out that Nucleai also has a suite of tools that are available for use with immuno-oncology products as well as multispecifics, and that these categories, while they are presented as discrete, there are overlaps between them that we'll talk a little bit about when we talk about the background of these therapeutics as well. Now, as the biomarker space and the therapeutics landscape is evolving, we have started to see more and more advanced types of biomarkers being launched within the space as well. And one thing that I want to point out is that this is not occurring in a single imaging modality or pathological toolkit. It's happening across the board. We're seeing more and more leveraging of spatial biology as a tool for understanding the tissue and the tumor response to these targeted therapies. So if we walk from the left to the right, what we've seen is, for example, AstraZeneca and Daiichi have published how to use a spatial proximity score to predict response to Dato-DXd in HER2-negative breast cancer. And that using that biomarker has a potential for enriching patients that are responders compared to those that don't respond as well to the drug using an IHC. Now, that's a lot of verbs. We're going to talk through all those very specifically. Don't worry, we'll get into that throughout the webinar. But in other modalities, such as multiplex chromogenic IHC, Nucleai has been part of a trial where we're using multiplex chromogenic IHC to specifically be an enrollment criteria for patient selection. And we'll talk a little bit about that as well. And then as you go into even higher plex, multiplex immunofluorescence is also entering the field of clinical development. And so across all of these different imaging modalities, we're starting to see proof points where the image itself from the tissue can provide biomarkers that are relevant to understanding a patient response. Now, this is going to be our first point where we're going to pause for a poll question just to get a sense as to whether or not people are considering biomarkers in ADC development and what types of biomarkers you might be thinking about using in that space. So we're going to hold this open for just a minute to let people vote, and then we'll show you guys the results from this. And while this is going on, one thing that we'll emphasize throughout the webinar is places where we have products that can potentially match to some of these technologies and the ways in which technologies are going to be used and leveraged to help understand ADC biology. So thanks everybody for participating in that. Lots of great responses here. It looks like there's a lot of folks looking across the board at both H&E and IHC, and that there are some specific other biomarkers that might be in play in more specific scenarios such as MIF or even RNA-seq. So thank you for taking the time to fill that out. I'm going to move on now to talk a little bit about Nucleai and the types of products that we offer and how those fit with mechanisms of action that are present within ADCs. So if we're talking about needing to improve the way in which we understand therapeutic response for antibody-drug conjugates, I think it's important to explain sort of our mission in this process. Nucleai is a company that really believes that AI-driven spatial biomarkers and diagnostics are going to be the way to better understand mechanistic response to these complex agents. We believe that powering pathology and enriching pathology beyond where it can be assessed currently is the way to provide the right type of context for matching the right patient to the right drug in this space. And we've been developing a suite of tools over the last 8 years or so, really geared around using any spatial biology platform as a way to interpret and interrogate the system of interactions that happen within a patient's tissue. So we can work with data from H&E, IHC, or multiplex IF, and we analyze that data with predefined algorithms that will classify and structure the tissue, both at the tissue level and at the cell level. And we'll talk a little bit more specifically about the types of models that we offer, but this is all really geared towards understanding very specific applications within the translational research space that's attached to ADC or IO development. And we work across the space to make sure that we can enable our pharma sponsors across the development journey of these drugs. We work with them for biomarker discovery and translational research all the way through potential companion diagnostic development. And we have several patents, indications, and markers that have been tested across the board with our models. So this is robust to the different types of tissues, the different types of markers that might be of interest for you. And we're working with over 60% of the top 20 biopharma in the space, including that prospective trial that I mentioned earlier. So I want to take a step back and talk a little bit about the mechanism of action of ADCs, and then we'll talk about the models a little bit more specifically that will help us understand how Nucleai develops ADCs and why we develop or ADC models and why we develop them. So just to level set with everybody, this is probably recap for a lot of folks, but I want to make sure we're all on the same page. There's a couple different major mechanisms of action that are primarily responsible for ADCs, but it's important to note that these vary by the actual specifics of the drug. So at baseline, an ADC is going to be an antibody that has some type of linker that attaches a particular payload to it. That's the broad classification. What that payload linker antibody combination does is it allows direct targeting of the tumor cell because it's got an antibody component that's going to sense an antigen on the tumor cell. And then it allows delivery of that payload directly to that tumor cell. What happens when you have that delivery to the tumor cell is going to generally be delivery of a payload and direct cytotoxic killing. So there'll be a direct mechanism of action where target cells that receive the payload die. There will also be indirect deposition of that particular payload to the general area that that cell is found in, either directly after the cell has been has apoptosed or has leaked out through some sort of release mechanism through the lysosome, for example. And so you'll have what's known as a bystander effect, where cells that may not be positive for that antigen can actually still receive that drug in a localized fashion. And then the third major mechanism of action potentially at play is through effector cells or other immune cell components, whether it's ADCC, myeloid-driven phagocytosis, or complement-driven cell death. And all of these components rely on the interaction of the tumor cell with the surrounding tumor microenvironment. So these are some of the key mechanisms at play. But this simplifies ADCs into acting on a single mode of action and a single framework. One thing to keep in mind is that there's a number of different types of therapeutic payloads that can be delivered. The primary ones that are used in most cases are either DNA damaging agents or mitotic inhibitors, and that there's a newer class of immune cell recruiting agents such as TLR or STING agonists that are also in play. And so you, again, have either direct cell engagement or direct cell death, or there's the potential for immune cells to play a critical role in the ADC function itself. And finally, the other component that we mentioned that's really critical here is obviously how you target the tumor cell. It would be remiss to just to gloss over the fact that there are many specific targets that have been identified for ADC development. There's probably a core of, I'd say, about 7 to 10 targets that currently form the majority of ADC targets in the market, but this is a space that's actively evolving as these targets move further and further through the phase, the clinical trial development stage. And you can think of this as, again, sort of allowing us to move from molecular biomarkers and immune biomarkers to spatially enriched biomarkers that are tumor antigens that are required for the activity of this drug. So here at Nucleai, the way that we think about using pathological data is to really start to break down what are the mechanisms of action in play and how can we model those with our AI models? How can we return information that's going to be specific to the mechanism of action? And so what we've done over the last couple of years has been to really focus on building models that address these mechanisms of action. These include target distribution and heterogeneity. So trying to understand from an IHC perspective, where is a target localized? How intense is the staining? And how much spatial heterogeneity do you have? And that can be both at the tumor level as well as the cell level, looking at where's the target distributed within the cell, on the cytoplasm or within the membrane, within the cytosol, or potentially in the nucleus itself. We also try to model the bystander effect. That's one of the things that spatial biology is great for, is actually understanding the physical localization of cells that aren't antigen positive and their relationship with antigen positive cells. And then beyond the tumor itself, beyond the tumor intrinsic signaling, we try to make sure that we can model off-site toxicity, trying to understand how many of the non-tumor cells express the antigen of interest, as well as immunogenicity by measuring how many cells are going to be interacting that are from the immune compartment and what types of cells are from the immune compartment. We're going to walk through all of these mechanisms of action that we can model with our data or with our AI models in just a minute. But I want to pause here for one more question. And this is really around trying to understand what you guys are interested in terms of understanding the mechanisms of action of ADCs. Which of these components is most interesting to you, and you would be most interested in modeling? This is going to be a single choice, so I know that you might have one or two that are tied, but we'd just like to know what of these you think might be of most value to you. And so as we get the votes in here, I'll try to make sure that I can emphasize as much as possible. We are going to talk about all of these particular components, but I do want to get a sense as to where we can spend and dive a little bit deeper, in addition to some of the questions that'll come at the end. Thank you all. This one's pretty overwhelming, so thank you for this. We'll spend quite a bit of time talking about target distribution and spatial heterogeneity. And we'll talk about some of the other components as well throughout the rest of the talk. So as we move through this, the way that Nucleai builds our AI models, again, we're platform agnostic in terms of the imaging modality, but we build models through deep learning through pathologist-driven annotations in many cases. And sometimes we do deep learning based on adjacent stains to try to augment what a pathologist might be able to annotate on a given stain itself. But our models are built to capture a couple key criteria. They're built to capture quality control and artifacts that it might exist within the tissue. These can be smudges, stains, folds of the tissue. They're built around then classifying the tumor into tissue areas, such as the tumor and the stroma, cell types, and then finally into standard and quantitative spatial biomarkers. And so these can be metrics that a pathologist can be benchmarked against as standard biomarkers, such as the tumor proportion score or the age score, or they can be features that go beyond what a traditional pathologist review is going to calculate, because they're focusing on interactions at a cell level or interaction at a structural level. And then after we return this data, after we build the model and work with this model, we return this through a consultative analysis process. So where we partner with our pharma sponsor to make sure that they're getting the most out of the data that's being generated. So to put a little bit of a finer point on this, what this looks like, we have a suite of generalized models that can help you structure tissue. Again, we break it into very specific areas, capturing the tumor and the stroma. We can capture various different cell level statistics, both the cell type as well as the staining intensity of a particular stain that has been used, as well as models that can potentially capture whether the staining is a partial staining or a complete staining. We work with our pharma sponsor to identify whether or not a specific type of information is important to their downstream hypothesis when we deploy these models. And then finally, there are segmentation models that allow us to capture optical density within specific compartments of a cell. And this allows us to interrogate questions related to target turnover, internal heterogeneity between different cells within the tumor tissue, and some metrics that may be related to the overall bioavailability of a given antigen. After these models are deployed, we allow you to access the data through our integrated viewer. We calculate our quantitative spatial features. And as I mentioned, we'll do a deep dive with all of our pharma sponsors to try to make sure that we are really driving to the hypothesis that's being validated with the project. And these can include things such as comparing OD distributions, subcellular staining relationships, or comparison with pathological assessments, where we provide external pathology review of both the model and scoring of the tissue with and without assistance from that model. Now let's talk a little bit about some of those mechanisms of action, and we're going to talk a little bit about where the field is today and some of the data that we can provide that help support these types of interrogations. So where I'd say where the most evidence is at this point in time in terms of understanding ADC response is around looking at tumor intrinsic signaling. This is looking for target availability of an antigen on the surface of a cell and looking at its heterogeneity in the tissue. I think one of the first stories that came out that really created a lot of excitement within the ADC space was thinking about how we could potentially expand some of the patient populations that would be potentially responding to therapeutic intervention in classically treated diseases. So, for example, in the HER2 setting, there is a case where as patients receive therapeutic intervention with an ADC targeting HER2, it became noted that there is a population of patients that didn't stain high by pathological review, but were responding potentially to the drug. And so there's been ongoing discussion about whether or not the particular IHC reagent was sensitive enough, or if we just didn't have the right tools to identify patients that were essentially very low expressors of HER2. There are rare cells within it that might be able to trigger a cascade of response, for example, and that those might just be missed by our standard pathological review. This has evolved in the ADC space. There's more and more biomarkers that are coming out, like I said, about the spatial proximity analysis that help further guide potential therapeutics in this space. And we're going to talk about how both of these types of scenarios can be addressed with AI models. So in the first case, when we're talking about HER2 staining, one thing that I think is important to baseline everything off of is the way in which pathologists are traditionally trained to score certain stains. HER2's been around for a long time, and HER2 positive patient samples have traditionally been from very high expressing HER2 cases. If you look at interoperator variability, and this is one example of a study that's been published in this space, but there's many examples of this. Pathologists are very good at scoring HER2 3+. There's very little interoperator confusion when you score this, but as you move into lower and lower values of HER2, pathologist scoring becomes more and more variable, and the impact of bringing in an AI becomes more and more beneficial because it allows for something that becomes more reproducible between pathologists. And I think of this as just sort of one of the standard cases where this type of technology is perfectly matched with a direct assistance need, where a pathologist is great at their job. They're very good at identifying these types of cells when they are common and when they're highly staining, but they might miss some when they are low staining or very light staining and very rare within a sample type. And so what we have seen is that we can take AI models to quantitate the number of cells that are positive at a very low level, present that to pathologists and help them identify patients that might fall into this, for example, ultra low status. these models are ones where we can directly tune this to our farmer sponsor needs in terms of what the threshold will be. And we can make sure that that model for where the intensity threshold is positive and negative, we partner with you on identifying. But this provides a quantitative readout of the number of cells that are positive for a given marker and breaks them down into their intensity buckets as modeled based on pathology training. And so this is one application where AI provides a pathologist a way to find that needle in the haystack. Which patients would you have otherwise missed that you can really think about potentially enrolling into an ADC trial because they might have benefit that would have been missed by standard scoring. The other type of scoring that we talked about was around spatial proximity scoring. And this is a suite of tools that isn't necessarily a fully defined list. It's thinking about spatial heterogeneity of a tissue and understanding what's the relationship between positive cells and their adjacent negative cells. So I'm going to walk you through this a little bit granularly and then give you some example data based on some work that we've done in the past. But for example here, what we're looking at when we think about spatial proximity scoring is thinking about whether or not a cell is close enough to an antigen positive cell to receive the ADC's drug cargo. So for example, there are ultimately three different things that we can tune when we create this framework for a spatial proximity score. We can tune the radius. How far away do you allow a cell to be? We can tune the number of antigen positive cells that it needs to see in order to receive that payload. And we can tune the intensity threshold of that antigen that's required in order to see that threshold. So this creates an AI model that has a wide parameter space that we can tune to the mechanisms of action specific to your antibody-drug conjugate, based on what you know about how the linker works, how the drug target itself works, and based on the staining patterns that we observe within the tissue. For example, we're showing a HER2 staining here where what we are looking for is the difference between positive and negative cells. And we're just doing positive and negative here for the purposes of illustration, but we can break this down into more granular information about the exact expression level within these cells. Anything that's shown in red is going to be those negative cells, IHC negative, and anything shown in purple is the IHC positive. So you see one lobe of this tissue right here, one group of the tumor core does have positive staining throughout most of it, but there are some tumor cells that group that are negative in this adjacent section. So if we think about this tumors, the standard pathological scoring for this tumor might actually end up with it being relatively low. Because I'm showing you a very zoomed in region, it's not giving you the whole picture of the tissue. But what we can think about is, okay, for those negative cells, how many cells around them are going to be positive and at what distance does that happen? And so for this particular cell, if we go out 30 micrometers, we don't view any cells in the direct adjacent tissue that are positive for that particular antigen. It's not going to receive an ADC via a bystander effect within a 30 micron distance. But as you move into 50 or 80 micron distances, you do start to see positive cells, and you would be potentially likely to receive that if the drug is able to diffuse that far within the tissue. And so what this means for that patient is even though pathologically they might score at only 16% positive, they might have higher and higher values for a spatial proximity score that could potentially be used to consider whether or not they should receive this antibody-drug conjugate. And what this means when you think about this, this is one patient, but if you think about how this might work within a clinical trial, what this means is that you might be able to enroll in, for example, in this case, maybe about 20% more patients if you use a spatial proximity cutoff rather than a traditional tumor positivity cutoff at the same value. Now, this illustrative example that I'm showing you here is one that has been very specifically focused on just tuning one of those variables, just tuning the distance of a cell between its a potential cell and the antigen positive cells that it's looking for. As I mentioned, there's two other parameters that we can tune. And as you think about the mechanism of your drug in terms of how many cells would be required and the exact expression level where you think binding is likely to happen based on what you know about the antibody component, we can actually model this a little bit more specifically for a given ADC. Now, that creates more complex relationships than the one that we're showing you here. This is a somewhat linear relationship, but as you explore higher and higher parametric spaces, you start to capture new structures within the data that wouldn't be readily available just using tumor positivity. And we work with our pharma sponsors to identify what are the critical components here, what's the type of heterogeneity that might be powering their response, and where might we want to be focusing some of the analysis of these AI models. But as we mentioned, this is only one of the mechanisms of action that we've got potentially on board. And there's other ways in which we can dive into this mechanism of action. But there are other ones that rely on the interplay between tumor intrinsic signaling and the tumor microenvironment. And we have a suite of tools at Nucleai that can help address some of these mechanisms as well. We have been working to build out toolkits that not only work on a single slide level, but can integrate multiple slides together so that you can virtually multiplex multiple different singleplex dab stains with an H&E or other combinations as necessary. And this allows you to take information that you might not be able to capture with just a single stain and drive it forward throughout the overall study. So if we focus there, we're going to focus on this first, and then we'll also talk a little bit about some of the multiplex IHC algorithms that we have available too. The reason I bring this up is because IHC is great for understanding on-target expression, but it doesn't give you as much granularity as some of the other models that we can provide. And you may want to combine different IHCs together to understand multiple different cell populations together. So rather than having to develop a complex assay, we can use multiple lower plex assays to understand different cell types. So for example, this is just PD-L1 staining with H&E, but you can look at tumor positivity, immune cell positivity, are traditionally the types of classes of cells that we're going to be able to capture with an IHC. With H&E, you can actually capture in more robust subtypes of cells, such as lymphocytes, granulocytes, plasma cells, because the hematoxylin stain has better definition of the cell structure for those cell types. And so if you needed to understand the relationship between tumor positivity and lymphocytic cell infiltration, rather than just general immune cell infiltration, a combination of these two assays may be more powerful than either assay on their own. And so we built a suite of tools that allows us to take monoplex assays or H&Es, co-register them together with accuracies of less than 10 microns across the slide, generally speaking, on average, and then look at where are these cells localized, what's the type of density that we see, and what's the very specific interactions that might be happening. In this particular publication that came out at Path Visions a couple years ago, we looked at trying to incorporate CD8, CD3, CD68, and CD163 staining to understand different cell types of both the lymphoid and macrophage lineages and their relationships to each other. But you can think about how this would directly relate to understanding immune cell components and their interaction with ADCs as you think about combining the ADC antigen IHC with the immune cell component that you want to model, whether that's through an IHC or with an H&E itself. And so these are a suite of tools that we can bring so that the trials that you already have in-house that have been running these standard assays, we can really add a lot of additional context to those assays and provide more information related to what the staining actually means from a biological and functional perspective. Now, sometimes you do need direct co-localization. You need to know whether or not two targets are expressed within the same cell type. And while we can estimate that from a virtual multiplex, direct detection of those two targets is going to be your best class best case for understanding that class of features. There are a few new ADC targets that are actually bispecific ADCs, for example, that use a bispecific antibody and then also deliver a payload. And so thinking about that co-localization or co-expression is something that a multiplex chromogenic IHC, for example, might be the right toolkit to leverage within this space. Now, as I mentioned, we work across all pathology toolkits, and one thing that we've found is that multiplex chromogenic assays are great for understanding co-localization, but sometimes they're hard to read and sometimes they're hard to quantify. It depends very much on the type of stain in terms of both the chromogens that are deposited and on the antigen, where the specific antigen is localized. If they're both cytoplasmic, if one is cytoplasmic and one is nuclear, or one is part of the cytosol. So it really depends on the specifics of the assay as to how much you might need assistance to help better quantify and understand the expression of these two targets. So the tools that we have built, in addition to our standard models that can classify cell types, we've built tools that help provide virtual DAB stains so that you can take a multiplex chromogenic assay and allow it to be more pathologically readable from a standard perspective. But we also have models that complement this that are learned to quantify the actual co-expression of these targets and classify cells into single positive or double positive classes based on the specific cell types that are going to be present with this assay. And this isn't limited to dual chromogens. You can use this with triplex or quadplex chromogenic assays as well. We work with the sponsor to make sure that the model is tuned to the assay that's in question. And so the last particular type of methodology that could be in play is thinking about scaling this up even further. We've talked a lot about IHC, H&E, and multiplex, but at the chromogenic side of the house, we haven't talked a lot about immunofluorescence. And this is a field that is both growing in terms of adoption in the research space, but also we're starting to see clinical trial engagement of these types of assays, usually in retrospective settings. But there are now examples of these types of assays moving into the prospective setting, like I mentioned at the start of the talk with that example with Akoya and Akravan working together to use these assays for prospective enrollment. Now, part of the benefit of these assays is that you can multiplex and ask questions across large numbers of variables, but they present a lot of hurdles in terms of the ability to QC the data, threshold it, normalize it, and get it ready for that downstream analysis. So a couple of years ago, Nucleai developed a suite of tools and a pipeline and platform for multiplex immunofluorescence analysis that we believe provides a generalized solution for understanding this data in an automated fashion. And when I say we believe that this is generalized, I want to emphasize that we've tested these platforms in oncology, I&I, and neuro spaces. We've seen that this platform works really well across these different disease areas, even though they have diverse markers, diverse cell types. We've analyzed over 3,000 samples and about 9 terabytes of MIF data at this point. And really, this is a platform that allows us to really work with any type of study for MIF data. Now, I'm going to dive into some of the specifics about what our platform does, but before I do that, I wanted to see if you guys are developing any spatial proteomics, multiplex immunofluorescence assays for your ADC research. We've seen some level of growth within this field, and we'd be curious to know if this is something that you are incorporating either into your translational research, or if it's something that you're incorporating into your actual trial as a retrospective, or even considering prospective use of these assays. And so we're just asking, do you have something in-house right now, or are you thinking about potentially developing this over the next year and a half or so? And after this question, we'll talk about what we offer in terms of capabilities for multiplex and some of the ways in which we can use it to understand both individual targets as well as combinations and ranking of different types of ADC targets against each other. Okay, so a lot of folks are thinking about it, not fully embedded yet, and that's probably what I expected here was that you're looking into this and thinking about where you might be able to deploy MIF. So what we've built in terms of this pipeline is a deep learning pipeline that takes multiplex immunofluorescence images, deconvolves them into their individual channels, and creates automated cell typing and phenotypic quantification of each of the channels that are present within the data set. What this means is that we follow a standard workflow where we do QC and inference-driven thresholding. We automatically normalize the tissue, identify individual cells, and create a robust cell typing framework that is specified based on the marker panel itself. And then we can build in further layers of information, such as neighborhood analysis and structural assignment of, is this part of the tumor core or the TME itself? This then builds on, as we build this out, what we have focused on is three key areas that we think is really important for this pipeline to be robust. First is on accuracy. When we've compared this to traditional clustering, we have a very high degree of accuracy, as you can see on the left here. And this is, again, compared to annotations that are completely blind to the training of the algorithm that were driven by pathologists that said, this is the type of cell that this likely is. How do we match up? Each point here represents an individual cell type. And as you can see, we significantly outperform unsupervised clustering type of analysis, which is relatively standard within the field. The second thing that we focus on is making sure that this is scalable. We want you to be able to bring in slides for a rapid turnaround if you need that for patient screening, or to be able to do this across large retrospective studies. And so we can take in slides from an individual patient sample and quantify the specific markers that are present, or we can do this at a much larger volume. And we're not limited to regions of interest. These are intended for use with whole slide images. And as we sort of highlighted on the past slide, it doesn't really matter which multiplex imaging platform you use. We can work with Lunaphore, Akoya, AltaView. Across the board, if you've got a multiplex image, there's a good chance that we can work with it in this framework. And as noted here too, we also allow the ability to register that H&E if you have it on a serial section or if you have a post-run H&E as well to provide further context beyond what the MIF might have. And then the last point is, again, we really wanted to make sure that this was generalizable. We don't want to build a model that's only going to work in one indication. We want it to be something that can work with different panel compositions, with different types of biology that you're trying to measure. And we've tested this on both low-plex panels, as well as panels that go into the 40s and 50s and 60s in terms of the number of markers on that panel. Now, I want to give you just one case study of how we think about using this might be beneficial in the high-plex space, for example. We built a panel of a 31-plex with ProPath about a year ago, and we've tested this on a sample of about 200 patient samples to look at several key tumor targets and their relationship with the immune microenvironment. And what we were looking for is trying to understand how do these different targets have various different characteristics that are related to different drug development strategies. And what we can capture with this type of panel is to think about different types of expression patterns. These can be common co-expressors, ones that might be beneficial for developing a multi-specific. They can be targets that are highly heterogeneous and that would be therefore better to develop two different drugs and combine them together to get better tumor coverage. Or they could be ones where you see consistent and relatively homogeneous expression across the tumor, and maybe the tumor core expression suggests it's a potential ADC. Each of these are types of examples that we see within these patients based on the different staining patterns that we observe. But rather than just providing you with a qualitative readout, what this allows us to do is to take it to the next step and actually start to quantify the relationships between these targets. So if we take a step back, how would we use this data to quantify and recommend targets for ADC development, for example? So here I've listed a number of targets, and this is within our colorectal cancer samples within this patient population. It's not the whole study. The rest of this will be showing at AACR this year, so please think about tuning into that. We'll give some more details about that in just a second. But if we look at the targets that are on the panel, one of the key questions was how much of the tumor is positive for that given antigen versus how much of the tumor might actually be seeing seeing a target if it had a bystander effect attached. So again, comparing sort of a traditional pathological score to an AI-powered pathological score. And so some of the traditional targets like HER2 and EGFR actually didn't benefit much from having a bystander effect added on top of the traditional targeting. Whereas other targets like cadherin-3 were variable enough in their expression that the spatial structure suggested that they would really benefit from a potential bystander effect based on this population set of data. Now, there are going to be some patients that express this target highly and some that express it lowly, but on average, what we saw was that cadherin-3 would benefit the most out of the targets that we were ranking. Now, you can also think about combinations of drugs and multispecifics as a potential way of thinking about combination, or just two different targeting drugs paired together when given to the patient. If we think about what this data provides us, it gives us another framework for thinking about rational combinations. One is around tumor coverage, where how much of the tumor itself would express either of the targets, but then how much of the tumor co-localizes those targets. So the first axis is one that really helps us rank the ability to do a combination, a multi-drug combination. And the y-axis acts as a way of predicting whether or not that combination should be one that is driven by multi-drug, like a target down here where there's not much co-localization, to one where there is a lot of co-localization, and a multi-specific would likely be able to cover the same amount of the tissue, but would have very limited off-target expression and off-target toxicity. And so these are some of the types of examples of the insights that can be driven by multiplex immunofluorescence data. As I mentioned, we will be at AACR 2025 this year. I would highly recommend that you guys consider popping in because we're going to be showing more data from this particular study. So please do check us out or drop by our booth. The information captured here will be present on our website as well. So we hope to catch you at AACR in Chicago soon. Now, we've talked a lot about the types of features that have been captured, and we haven't talked a lot about the process by which we engage with our customers throughout the life cycle. We obviously can capture and structure a ton of different types of features related to individual biomarkers, and a lot of this is really useful in retrospective analysis. But we've also got experience working with deploying these types of algorithms, not just for use retrospectively, but also planning them out for commercial companion diagnostic development. And so there is a three-way partnership that we have with one top 10 biopharma and top 3 IVD partner, where we are working with using our algorithms in a prospective setting, where we do need to return that data in a very robust and reproducible fashion, in a very time-bound fashion due to the clinical trial environment. The way that we set this up was we ran sort of a 3-stage approach. We created a feasibility for the specific model, ran a prevalence study like you would do with developing the actual assay itself, and deployed a QMS-regulated model that has been validated in parallel to the assay for the clinical trial itself. And so we built out a framework for not just building these algorithms, but capturing the regulatory requirements and reporting requirements necessary to implement these into clinical trials. And what this looked like from a functional workflow perspective is that the samples, as they move to the central testing lab, are stained, imaged, and transferred to Nucleai for its image processing and algorithmic scoring. This is then transferred back to the CLIA lab, where a pathologist reviews and agrees or disagrees with the score that's been given. They have an opportunity to overwrite that if needed. And then that information is sent back to the clinical trial site. And all of this happens within the course of less than 24 hours for our algorithm prospect, our model deployment, so that we don't interrupt the clinical workflow of the trial itself. And this is powered because we offer not just specific model technologies and not just specific services related to analyzing your data, but also because we built out QMS and regulatory components, and that we have relationships with key labs and diagnostics specifically across the board. And so in this space, I guess the last question that we want to ask you is, as you think about your ADC programs, are you considering additional AI augmentation beyond just pathological scoring? And this can be in retrospective or prospective settings. But we'd be curious to understand how we might want to fit within your framework for evaluating these clinical trials. So as we wrap up this poll, this is going to be the last question that we ask you. Looks like most of you are here for a reason, and we're really glad to have you joining us. We are definitely interested in understanding what your needs might be. We'd encourage you to reach out if you want to follow up on more specific applications that are related to ADCs or any of your drug targets that are in your pipeline right now. But thank you so much for spending time with us today to talk about some of the solutions that we provide in these spaces.
Thank you, Jason. As always, a really articulate and wonderful talk. So we'll now move on to the live Q&A portion. As a reminder, if you have questions for our speaker during the live Q&A, you can use the Q&A box at the bottom of your screen and type it up. So Jason, we had a few questions that started coming in during the talk, so we'll start with those.
Perfect.
The first one is related to the wet lab side. Are there any requirements for the scanner resolution or magnification for the AI scoring models?
So we routinely take in either 20x or 40x imaging samples. So we can work with either of those. One thing to note, obviously, is that certain models perform better with higher resolution. The more granular you want to get on segmenting certain cell types, you might want to consider working with a 40x scanner, but we can help provide guidance on that as we're scoping out the project when we're considering that with you. And if it's necessary to do some rescanning, we can help connect you with labs that can do that as well.
Wonderful. The next one is a little more clinically oriented. What evidence have you seen about the correlation between novel computational scores and clinical outcomes for ADCs?
So we touched on some of this. This is an evolving space right now. AstraZeneca probably has the largest body of evidence suggesting the need for AI augmentation within the ADC space. They've been pushing an evolving suite of tools that they call quantitative continuous scores that are spatial features that capture various different interactions at the individual cell level and subcellular level. These have been toolkits that have been evolving and evidence that has been growing over the course of their phase 3 trials that they've been watching. But we are seeing a lot more interest across the board. And I suspect that this year and next year will be ones where there will be a lot more information coming out related to the types of biomarkers that are going to be validated. And we are seeing a lot of interest across the board in understanding the mechanisms at play between these types of spatial biomarkers.
Right. Now, something related to the markers, maybe on the wet lab side. How flexible is it to replace markers in your existing multiplex platform? And how long will it take to develop a new platform? Yeah.
So on the multiplex side of the house. So for example, for our multiplex immunofluorescence pipeline, The way that we approach building out the outputs is thinking about the markers that are present on the panel and thinking about what that tells us about which cell types we can accurately classify and which markers we can accurately quantify within that ecosystem of data. Honestly, this is a really flexible system. If you swap out an assay from one particular type of readout to another, one particular panel to another, we're not going to be the limiting type factor here in terms of a time to building a new model. It's going to more be related to validating that new antibody that's part of the MIF panel. But on our side, the analysis is pretty straightforward to change what the cell rules mean when you move from one panel type to another. So we routinely work with folks as their panels evolve, as their solutions evolve, and recommend changes that could provide them with further granularity. We've got in-house experts that think about optimal panel design and potentially the ways in which you could tune a panel to be more specific to a specific question. We'd highly recommend engaging early on in the process because a lot of platform vendors that are going to be validating these panels are thinking about them from the perspective of, does it make a nice, pretty image? And we really encourage you to think about also, does it provide you with the dynamic range that's necessary to capture your cell type of interest? And we can help with that process. So if you're interested in building a panel, if you don't have one yet in-house, we're happy to provide some consultation at those early stages to make sure that the data that you get out at the very end is going to be driven towards success as a whole.
And maybe a slightly related question. How scalable are your ADC models across different stains and indications?
Yeah, so we built these models to be relatively generalizable, but we do QC every model that we work with. So one thing to note is that each particular antigen is going to have a slightly different staining pattern, and each particular antibody and protocol for staining that antibody is going to have a different staining pattern attached to it. So while the models are generalizable, during the course of evaluating it, during your trial, during your project with us, we build in robust QC metrics where we take and compare this both to pathological assessment as well as to computer vision assessment to make sure that what we're providing to you is the highest quality possible. And so our services that we provide, when we return a model to you, you'll have a direct understanding, we'll have a direct readout of what was the accuracy of the model, how close to our acceptable thresholds is that, and we flag samples that have, for example, too low tumor content to be analyzable with the model, or ones where the artifacts are particularly high. So you'll have information both at the study level, understanding the QC of the whole study, as well as individual slide level QC. But we've not found a particular antigen at this point that we can't work with. At this point, if you've got an IHC stain and you want to capture some of these particular features, we will absolutely work with you to get the most out of that data.
And I presume on the MIF side, that's been a lot more generalizable, I suppose.
I mean, again, it comes down to validating the panel and making sure that the antibodies are performing as expected. But if they are, then these types of metrics can be quantified across the board there.
Wonderful. Another question is, have you also worked with RNA spatial data in the context of ADC biomarkers? And I wonder if we could also comment on this when we think about RNA and protein and yeah.
Yeah, so there's a little bit of different information that you get with RNA and protein. RNA, generally speaking, you're either going to be talking about something like RNA scope for a low-plex assay, or you're going to be talking about some level of spatial transcriptomics to get more of a broad view. We have worked with both of these types of data in the past to some extent, but they're not our bread and butter, mostly because a lot of the statistics that we are finding are really critical in the field right now are antigen driven, right? If you want to understand target turnover within a cell, you really need protein expression to that. And if you are analyzing data that is from RNA, you need to validate that the RNA quantification actually matches the protein expression down the road anyway. So we have done a little bit of work in those spaces. We have models that are focused on some of these projects, but they're not what I would say are routine asks in the field. But if you have a project within that, please do reach out. We'd be happy to scope it with you and see if it makes sense for us to be the vendor to partner with on that, or if we can provide recommendations on how you might consider folding that into a broader biomarker strategy as well.
One more question, and then we'll have to unfortunately wrap up, but we can always reach out and answer questions directly one-on-one with our attendees. How did you select your antibodies for target detection?
So for the MIF panel that we described here, this was developed a couple years back, and we specifically were targeting a combination of two different things. We wanted to capture a couple different ADC candidates that were in the pipeline that were relatively common at that point in time. We also wanted to capture targets that were being considered for multi-specific development as well, because we wanted to have this be a benchmarking data set that we could compare the overall landscape of what was happening within the oncology field, which captured both tumor intrinsic signaling as well as the immune biology. When we think about the right way to develop some of these panels, it's really important to think about what hypotheses you really want to test with them, and what cell types are critical to answering those questions, and defining the content based on those. Once you can quantify the cell types that you're interested in and the particular antigens, then you can start to get into a lot of the nuanced interactions that we talked about today and benchmarking those against each other. But that was some of the rationale that went within that specific panel that we developed. But I did focus on the ADC component a little bit specifically in this talk.
Wonderful. That's all we have time for today, folks. In 4 weeks, we will be at AACR, so hopefully we can meet up in person. We will have more data on the posters we're presenting, as well as exciting booth demos. And if you can't make it to AACR, we have the next Nucleai Academy, which will come up in about 6 weeks or so. So with that, we're going to wrap today's session. Thank you so much, Jason, and thank you to all our attendees.
Thank you. Have a nice day, everybody.